Hello and welcome to our next video on our venture through biology. We are in a chapter in Clark, Choi, and Douglas, Biology 2E, out of Rice University, open source. Um, in this chapter 5, we're talking about the cell membrane, and we're talking about how things get in and out of the cell. And so we want to talk about this. I've divided up this section into two parts. This first part is on diffusion, channels, and carriers. So there's a section in this chapter on passive transport, and there's a section in this chapter on active transport. Passive transport, what is, you were asking, what is passive transport? Why, passive transport is where stuff comes in and out of the cell, and it doesn't require any energy. The cell doesn't have to be plugged in for this to happen. It doesn't take any energy to accomplish this. It's passive transport. It happens just because of the physical laws and such. And so the cell membrane, first of all, is selectively permeable. What that means is it doesn't let everything in and it doesn't let everything out. So we talked about there being bouncers on the surface of the cell saying, I'm sorry, you're not 18. And so the cell membrane is selective. It's selectively permeable. Permeable means stuff coming in and out. And so passive transport is where stuff comes in and out and nobody has to do anything. Nobody has to charge it. You don't have to plug it in. This just happens by the mystery of physics. For example, the movement from a higher to a lower concentration. Now, let me take this expression, concentration gradient. What is a gradient? Well, a gradient is basically like if you're going downhill, you're driving through Tennessee and you're going from Nashville to Chattanooga and you're going downhill. That's a pretty steep gradient. And they have those truck runoffs, right? So a gradient it doesn't have to be steep. It can be a slow gradient. But gradient basically means that there is a difference in amount from one end to the other. So for example, let's say you have a higher concentration of something outside of the cell than you have inside of the cell. Well, basically over time, that's going to even out as much as possible to where they become equal, where we get what's called dynamic equilibrium. Dynamic equilibrium means that, yeah, there's still some stuff coming in, and yeah, there's still some stuff going out, but it's happening so that it remains pretty much the same concentration outside and inside. This happens with pressure, as well. But anyway, so movement from, say, a higher concentration outside the cell to a lower concentration inside the cell is going to kind of passively suck concentration outside to the inside until it reaches equilibrium. So that would be an example of passive transport. Doesn't have to be plugged in, it's going to happen. In the same way that okay, well, let's just be crass. Why not? If somebody farts, initially there is a high concentration, around a certain part of the anatomy, but it spreads out, and eventually the bad smell is evenly distributed throughout the room, and of course, eventually it diffuses into the equal atmosphere to where we can't smell it anymore. Although your mother, Sophie, has a very good nose and can smell it longer than I can. Curses. Anyway, so this movement from a higher concentration to a lower concentration, it's very natural. And it's an example of passive transport. It's an example of diffusion. So hold that thought about diffusion. Diffusing farts. Hold that thought. Before we get to diffusion, let's just remind ourselves again what it means for something to be selectively permeable. So part of what makes it selective is the fact. Remember, our phospholipids are very attractive balls with legs. The ball part of the phospholipid is polar. And therefore it's attracted to ionic stuff. But the legs of the phospholipids are lipid. They are nonpolar. And so basically, the phospholipids facilitate nonpolar things getting through, because that whole middle part with the legs, legs on legs, that middle part is nonpolar. And so nonpolar stuff can get through, but polar stuff can't get through. Um, and so the reason that the cell membrane is selectively permeable is because you have that nonpolar middle part of the lasagna. By the way, ambiphilic means both philic. It means both, liking both. And so I'm not sure it's a great term, but it basically means that the phospholipid is both hydrophilic on one end and hydrophobic on the other. And so the fact that it is both means it's ambiphilic. I don't like the term. Well, I don't have a problem with the term. It just isn't very easily illustrated. Okay, so this is what I've just said. Nonpolar and lipid-liking molecules, that is, lipid-soluble molecules, can make it through the neutral zone. By the way, so I fried hamburgers tonight, and let's just say that it's hard to get grease off your hands. It's hard to get the fat out of the pan with water. Why? Water is a polar molecule. Fat is a nonpolar molecule. And so, it's just difficult to get the water to wash it away. Anyway, in the same way, nonpolar likes nonpolar, and polar likes polar. And so that nonpolar and lipid-liking molecules, okay, you're good, come on in. They can make it through the neutral zone. But polar ions and polar molecules have a hard time. We didn't really talk about polar molecules, but just ignore me for now. Okay, that's a little bit on selective permeability. Back to diffusion. So again, here's a nice illustration. Thank you, Clark, Choi, and Douglas, Biology 2E, for this little diagram. So this is the lasagna membrane, the cell membrane. We've got the balls and the legs standing on top of each other. And so here's an example of diffusion. So this is time. So we start off with a high concentration outside the cell membrane. Well, eventually, these are going to diffuse like a fart in the air. These are going to diffuse into the cell, into the cytoplasm, until eventually we have a dynamic equilibrium between those outside and those inside. So this is an example of passive transport. Doesn't have to be plugged in for this to happen. Movement from higher concentration to lower, and dynamic equilibrium is the end game. That's where it's all headed. Diffusion. Okay, so factors that affect the diffusion rate. How quickly does this happen? Well, how big is the gradient? Again, the gradient is the difference, like going down the hill. So the bigger the gradient, the bigger the difference in concentration, the faster the diffusion is going to happen. The mass of the molecules. Slow molecules move slower. I can't get into the cell. I'm fat. Well, I'm of large mass. Sorry, I hope nobody was offended by this video. It's just to help us remember. Temperature. So the bigger the temperature, the more the molecules are moving. And the more the molecules are moving, the more they're going to get in. So temperature increases the rate of diffusion. The density of the solvent. So if we don't have a lot of water, then, well, the more dense the solvent, it's crowded. And so I'm trying to get in. I can't get in because the liquid is too dense. I can't get out. The liquid is too dense. So the denser the solvent, the slower the diffusion rate. And this is what happens in dehydration, because without water to make things flow, then it gets all crowded. And it makes the diffusion not happen very quickly, which means the energy cells don't get what they need. And nerve cells are particularly susceptible to this problem, which is why you pass out and go into a coma sometimes with dehydration, because your nerve cells basically can't function even sooner than other cells in your body. Solubility, we've already mentioned this, that the polar, nonpolar stuff affects the rate of diffusion. Cell size. So the bigger the size, they run out of energy. I'm getting tired. I can't get where I need to go. It's just too far. And so the cell size, the bigger the large cells can't survive. And of course, I'm throwing this onto this slide. That's the end of that section, but the end of diffusion mentions filtering. Filtering, like the kidneys filter stuff. Pressure helps. So in the kidneys, there's pressure pushing stuff through, and high blood pressure, of course, can be detected by, if like you have proteins in your pee. It's not supposed to be there. It's because there's too much pressure. It's pushing it through. It's not supposed to push through. Solutes are things dissolved in the liquid. Okay, that last one could have been put on a different slide. I'm sorry, I squeezed it into this one. These are the main ones affecting diffusion rate, but pressure does affect the filtering rate in things like a kidney. Okay, transport proteins. So these are, what are they called? Facilitator proteins, but I'm going to call them transport proteins. So we've got a problem. How can the polar ions get inside of the cell? I mean, because we've got these very attractive phospholipids that are telling the polar bears that they've got to stay out. And so how can the polar bears get into the bar? I mean, into the cell, into the polar bar. How can it happen? Well, we have a couple proteins that, remember, these are called integral proteins. The integral proteins are the proteins, they span the whole of the membrane. And so this is called a channel protein. And it's mysteriously called channel, I think, because it's a channel. Think of it as a slip and slide. Woo, woo, in the channel, it's the channel slide. And so the ions slide through the integral protein into the cell. And they're like, nah, 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 because these hydrophobic legs are trying to keep the ions from getting in. But there's a nice little slide that the ions can get through in those channel proteins. And so they shield these ions from these phobic, fearful, these high water fearful legs. And so that's a way for them to get through. Channel proteins. There's also a group there's a group of these called aquaporins, which is an interesting name. It lets the water pour in. And so some of these are like water. Come on, water, come on, because water, hydrophobic means afraid of water, right? And so the water can get through certain proteins called aquaporins. Now, some of these proteins have gates, that they can let some in and not let others in. The chapter suggests that nerve cells, I believe, especially can have gates to allow certain ions in and out. Of course, the way that and muscles also have some of these, because this difference in charge, helps move my muscles. That's an ionic thing going on based upon differences in charge. And so the gates kind of allow ions in and out according to the plan. So anyway, transport proteins, number one, channel proteins. Channels only. I don't remember the rest of the song, but this is the kind of protein that this is. Channels only. Okay. The other kind of protein are the carrier proteins. And I'm sorry I didn't let it animate. There we go. The carrier proteins are not slip and slides. They're an escort service. Again, forgive me. They escort certain molecules through. They can actually, they change shapes to squeeze things through. They are carrier proteins. There are all kinds of really bad metaphors I could use here that I'm not going to. I've had at least two completely inappropriate metaphors come to my head. Anyway, I'll let you whatever you use to remember these, God will forgive you, because it's for a biology test, right? So it changes shape to squeeze whatever it is through, and these can be selective for a single substance. So you can have specific carrier proteins that only let, I'm just making this up, a chlorine through. Only chlorines can, and I don't know if that's a thing. It might be. But so you have carrier proteins that just take specific ones. Okay, so what have we talked about in this video? We have talked about, this is the first part of a section on passive transport. What's passive transport? Passive transport requires no energy for it to happen. It just happens because of differences in concentration and so forth. Diffusion is, again, this passage of things through the selectively permeable membrane. We mentioned a number of factors in how quickly the diffusion rate is. And then in the last part of this video, we talked about transport proteins, namely channel proteins and carrier proteins, both of which are integral proteins that span the whole of the cell membrane and the bilayer of phospholipids. This has been passive transport through the cell membrane, part 1.